Introduced to Australia to control agricultural pest species, cane toads became an invasive pest themselves. In 1935, the Bureau of Sugar Experiment Stations brought 102 cane toads from Hawaii to the Australian state Queensland with the intention of managing 2 beetle species, of which the larvae are major pests in sugarcane plantations. However, the attempt using cane toads as biocontrol agent failed, as the toads did not effectively control the insect pests. However, the toads found enough food in form of native animals and multiplied and spread quickly across large areas. Due to their toxin, cane toads have only few natural predators in Australia. Native predators unfamiliar with the toxins, such as some reptiles and mammals, are now at risk when eating cane toads. As a consequence, cane toad populations increased rapidly, while those of native predators dropped. Today, cane toads are found all over the north and northeastern parts of Australia. We spoke to one of the first researchers to discover the ecological effect of cane toads on Australia's native fauna, Professor Rick Shine from Macquarie University in Sydney, Australia. Among many other achievements, Rick's research focuses on 2 key topics: spread and rapid evolution of cane toads, and conservation of reptiles and other large predators. At one of his research sites near Darwin, Rick conducted a major research program on reptiles. When cane toads arrived in this area, he saw an opportunity and began studying the effect cane toads have on Australian ecosystems. Rick is considered the world's leading expert in cane toad ecology.
Rick, in the 1930s, cane toads were introduced to Australia as biological control method, but it failed ultimately. You were among the first to discover their harmful impact on the native fauna of Australia. How did you first realize the extent of the damage they caused here?
Yeah, well, there'd been stories around the place of, the big goanna found dead and things like that, but I think people didn't really care very much about reptiles and amphibians and animals like quolls that ate your chickens. They cared about big, beautiful birds and mammals, and the toads weren't affecting them very much. So, I guess we didn't really know. I'd heard these stories, and we were in this fortunate position of having studied the fauna of the tropical floodplain up near Darwin for 20 years. So we knew what was there, and it became pretty damned apparent as soon as the toads arrived that we had the bodies of goannas lying all over the ground, and the numbers of some of these species just plummeted. And it was pretty damn clear what was going on.
So, and then in the aftermath, you proposed spatial sorting as an additional process of evolution, also with the example of cane toads, alongside natural and sexual selection based on your observations up north. Can you tell us what this means, the spatial sorting, and how it actually works?
Yeah, so one of the first things we did when the toads arrived was to start looking at how much they moved and so on. And we discovered they were moving vast distances every night. When we'd radio track wild toads, they were moving 1 kilometre or so. Whereas work that had been done on the Queensland coast suggested that the toads were just moving a few metres and were very sedentary. And so we started looking at what was going on, and everything we found out about toads suggested that the ones at the invasion front were incredibly different animals from the ones further back in the long colonised areas. And the guys at the invasion front were incredibly dispersive. In the end, we bred those animals and we found that their offspring inherited those traits as well. So we had evolution. And the question was, what was the mechanism? Natural selection was the obvious answer. But there was another answer that had kind of been hinted at in some mathematical models. And that's a simple sorting of dispersal enhancing traits in the course of an invasion. Essentially, if you've got genes for being slow and not moving very far, you get left behind. The only ones at the front are the guys that have got genes for being really active and fast and continuously moving. When it comes time to breed at the invasion front, those guys breed with each other because they're the only ones at the front. We call that the Olympic Village effect because the athletes are interbreeding with each other. Now, some of their offspring have got genes for long legs from dad and genes from endurance from mum, so they go further than either mum or dad went. And it's a cumulative process. Every generation, any gene that makes you go faster ends up at the front. Any gene that makes you go slower gets left behind. And so you evolve a totally different kind of an animal simply because of differences in rate of dispersal. There's no difference in lifetime reproductive success. There's no fitness differentials. It's simply driven by how quickly you can move across the landscape.
That's fantastic, yeah. So where are we now in cane toad control, also all this considering? Is there any hope to halt the spread of cane toads? And is there also any chance for us to help native Australian wildlife here?
There's always hope. Toads have been a really difficult problem because they have such incredibly high reproductive rates. Tens of thousands of eggs in a single clutch. So you can't just go out and start hitting cane toads on the head and think you're going to make a difference. What we discovered is that many of the vulnerable predator species can coexist with toads. If the first toad they meet is a small one, they eat it, they get sick, and they don't eat them again. They learn. It's called conditioned taste aversion. Any of us that have had the wrong thing to eat or drink and have become sick afterwards know how long we remember not to eat or drink that kind of substance. So the big threat from toads is that they're very poisonous. As long as you don't eat them, you're fine. You can live with them. And so one of the things that we've done recently is start releasing small cane toads in advance of the front. That way the predators, like the big goannas and so forth, encounter a small toad first, they eat it, they get sick, they learn, and when the big toads arrive in a month or two's time, the goannas leave them alone. And that's looking really encouraging. It's looking as if we really can save predators across a huge area. If you can just save them for that first year or so, the toads arrive, they begin to breed. There are lots of baby toads around to teach the newly emerging baby predators. So a single generation of education might be enough to keep some of these endangered predators in the landscape despite the presence of cane toads.
Rick, you can look back at a long and super successful and fruitful career. Can you share with us what keeps you motivated? Why are you still going and be as research active as ever?
Yeah. I think it's simple childish enthusiasm. As a kid, I loved reptiles and amphibians. I thought they were fascinating. I encountered natural selection and those sorts of ideas when I was in high school, and I just thought they were phenomenal. So simple, but yet with such incredibly complex ramifications. And I just feel incredibly privileged to have spent my lifetime finding out answers to those questions about the animals that I find so fantastic. So, yeah, look, I don't have a good explanation for why my enthusiasm stays as high as it's always been, except that it's just so goddamn much fun.
So if you look back at your career from starting in the 1970s up to today, academia changed a lot. So what do you think are the greatest changes in academia since you really started out first?
It's evolved faster than cane toads. It used to be a very different world. There were far more resources. When I was an undergraduate student, every academic would be given a full-time research assistant. Those days are long gone. We've had an avalanche of paperwork of various kinds that have made it much more difficult to do the admin, and time has become so precious. Even early in my own career, academics would all gather around at morning tea time and sit around and talk for half an hour about their research and maybe about the football, but everyone felt like they had spare time. And these days, everyone's under the pump. And so a lot of that collegial discussion has disappeared. One of my joys of being at Macquarie Uni is that it has hung on here far better than in any other institution I've been part of.
Rick, over your lifetime also as an active academic in Australia, you probably have seen the most amazing fieldwork experience and so on. Can you share one very memorable moment with us that still sticks to your mind?
There's too many of them because every time you go out in the wild, unexpected things happen. But I was thinking about this, and I think probably one of the most memorable was at Fog Dam, our field site in tropical Australia, where I've worked for almost 30 years now. There are crocodiles in the dam, and including some very large saltwater crocodiles, but you get to know them, and you know, they're not a problem. But during the wet season, the big salties can move around. And one wet season trip, Greg Brown and Melanie Elphick and I were wading through the water up sort of knee deep in the middle of the night, testing foraging responses of snakes in the water, when suddenly this huge saltwater croc that we'd never seen before turned up and just appeared on the water surface about 20 metres away. And we immediately knew he wasn't any of the local guys we're familiar with. And he came roaring towards us on the surface of the water, which was weird. He wasn't creeping up to attack us, and I have no idea what he was on about. But it was sort of bowel loosening because there's this giant croc on the surface of the water, making a bow wave, racing towards us. And I'm starting to think, you know, middle of the night, we're knee deep in the water, you know, exactly what do we do? And Greg Brown, who's my long-term postdoc, reached back, pulled a slingshot out of the back of his jeans, got a rock out of his front knee, and just went ka-thunk, and at about 10 metres hit the croc on the side of the head, and it reared up in the air, splashed and roared off, got the fright of its life. Greg had never had a slingshot with him on the hundreds of times I've been with him in the dam well before. And I said, why did you bring that? And he said, I just had a feeling tonight might be the night. That was probably one of the more memorable times.
And we are lucky that we still have you here. Thank you so much for the interview, Rick.
My pleasure.